everyone. Welcome to your genetics lab. My name is Dr. Jones, for those of you who haven't had an opportunity to meet me yet. And today what I'd like to do is give you a brief video introduction into what our online laboratories will look like, and then give you some background information on this week's online laboratory, which will cover the techniques of polymerase chain reaction and gel electrophoresis. These are two techniques that are very important to the study of genetics. They are things that you will have the opportunity to do in a hands-on in-person lab. So this online lab is really intended to give you some familiarity with these techniques before we actually do them in person. But before I talk to you about PCR and gel electrophoresis, what I'd like to do is give you a little information on the structure of our labs for this semester. So what I have for you here is a schedule that is the exact same schedule that I've published for you on your syllabus. By the way, if you need a copy of the syllabus, you can find it in the email that I sent out last week. If you have lost that email or can't find it, I also have the syllabus posted for you on Blackboard, and I'll show you where to find that in just a moment. Okay, so this is our lab schedule. Let me point out a few things. First of all, you'll notice that I have the weeks here, and I've just put the first day of the week here, the week starting January 19th or January 25th. So I've only put the Monday date, but please know that this schedule also applies to the Tuesday lab as well. So for example, this would read 25th/26th, 1st/2nd, 8th/9th, etc. So I just put the Monday date here, but the same schedule, of course, applies for the Tuesday labs as well. The next thing that I want to point out to you is that the activities that are in bold are the activities that we will be doing in person, okay? So in this case, what's happening is on the 25th or 26th, my lab group A will be in person, and you'll be doing a lab exercise where I show you how to do gel electrophoresis and we construct some pedigrees. Now, if you're in my lab group B, you will be online this week, okay? And you'll be doing something called a Labster simulation. The Labster simulation will also cover gel electrophoresis and the polymerase chain reaction. Now, notice that these lab exercises occur in 2-week increments. So here's a 2-week pod, here's a 2-week pod, here, here, and here. So what's happening is group A is in person 1 week while group B is online. The following week, we simply flip-flop. So the same activities are being done by both groups. We're just moving back and forth. The significance of that for you is that in order to avoid confusion, all of the assignments from either the in-person or the online components of this 2-week pod will be due at the end of this 2-week period, okay? Or actually right before we start the next thing. So your due dates are posted for you on Blackboard, but just so that everyone knows, this first set of labs will be due either on February 8th at 12:30 if you are in the Monday lab. And if you are in the Tuesday lab, this first set of activities will be due February 9th at 12:45. In other words, we're going to go through these 2 weeks. All of this stuff has to be done before you start the next group of labs, okay? Again, if you have any questions, these due dates are posted for you on Blackboard. I also want to point out, if you're unsure whether you belong in Group A or Group B, I did send an email out detailing that for you. And if you can't find that email, or perhaps I had your address incorrect, you can find this information on Blackboard. And again, I'll show you where to find that in just a moment. There are a few times you'll notice when we are not in a 2-week back and forth kind of construct. So here, this week, both Group A and Group B will be online, and that's because that's what we call a Patriot Pause week, where we will have a day off during the week. We have another week here where both groups are online, and we had to do that for scheduling purposes as well. And then finally, there's one last time where we will all be together, but in this case, we will all be together watching presentations that that lab groups have made detailing a genetic disease. And I'll be telling you more about these presentations later in the semester. Now, I want to talk to you in a little bit more detail about the online lab components. So just those things that are not in bold. You'll notice that I have them called out in 2 different types of activities. I've got some things that are online that I'm calling Labster simulations or Labster sims. So I'm just going to put Labster here. And then I have a second set of online things that I'm simply designating as online activities. So these are things that will not involve our Labster software. So let me talk for a second about the difference between these 2 online remote laboratories, okay? So both a Labster assignment and an assignment that I've designated as an online activity, these are both online or remote, okay? You're not going to come to class on weeks that these are assigned to you, okay? Online activities are simply exercises that myself or Dr. Bauer have authored. These typically involve some sort of web research, or they might be a set of problems that you'll do independently, okay? So for example, one of our online activities that Dr. Bauer and I authored last semester involves looking at the infection rates of COVID-19. We're going to learn a little bit about the COVID-19 vaccine. Another online activity will be looking at what's called CRISPR gene editing. But then you'll also have some problem sets where you will be assigned pedigrees to create for families, or you may be assigned some genetic problems, okay? If it's a week where I tell you that you're doing a Labster simulation, that is essentially a piece of online software that FMU has purchased that allows you to, in sort of a video game environment, conduct a virtual lab activity. And I'm going to show you how Labster works in just a moment. Now, regardless of whether it's what I'm calling an online activity that Dr. Bauer and I authored, or if it is a Labster video game activity that you're being assigned. Regardless of whether it's either of those, I will always publish for you a video tutorial for each online lab. So these tutorials will talk about what the assignment is, give you some background information on the assignment, maybe give you some tips on how to complete the assignment, and also point out for you the due dates. So you'll always know what's going on. Obviously, if you still have questions after viewing the video tutorial, you're certainly welcome to contact me via email, phone, when we're in person, and you can also contact Dr. Bauer because we are teaching very closely this semester. Okay? Now this week, what you have been assigned is a Labster activity. So you're going to be working in this online sort of virtual video game environment, and you're going to be conducting an experiment using the polymerase chain reaction. So what I'd like to do is to show you a little bit about how the Labster platform works. I'm going to change my screen share. And hopefully I have done this correctly. And what you should now see is the home page for our Blackboard class for the lab. okay? So this is actually the Monday Lab homepage. And what I want to point out to you, first of all, I don't really use the homepage a whole lot. The one thing that you will want to pay attention to on our homepage is the announcements tab. Generally, I will both send you an email with announcements, and then I'll duplicate the same announcement here in Blackboard. And I do it in both places simply because this semester, there's a lot of email traffic because a lot of classes are remote. And so I find that lots of people are losing emails, things get buried. So in case you can't find an email, you can always capture the information that I've sent to you by clicking here. So here you can see a copy of the email that I sent out last week. This is the Monday group, and there's information on which people are in group A, which people are in group B, and where to find all the information you need to complete your labs. So that's the home page. Where you'll spend the majority of your time is on the content tab, where I will be publishing things like your syllabus. If you click here, you can find the syllabus. You can find information on the Zoom office hours that I have. And then you'll also find folders where I'll post all the information for our online activities. So here you can see the Online Lab 1 folder. You click here. There's links to 2 different Labster activities, and there will be, after I finish this video, a link to the video as well. So these Labster links, all you'll have to do is click. You're automatically enrolled in Labster. There's nothing for you to purchase. It'll take you directly there. The introductory PCR is what you'll do individually. Today, we're going to go through this Labster demo together so that you can see how the platform works. So I'm going to click on this, and that automatically takes me to the Labster simulation. Now, I have already previewed this, so it's asking me if I want to restart this Labster, and I'm going to say yes. But before I click on that, I want to give you a few tips about using the Labster platform. First of all, Labster works best in either the Google Chrome or the Firefox web browsers. It does not work very well with Safari. So if you happen to have a Mac, I recommend using Chrome or Firefox. Safari will mess you up. The other thing that typically happens is it takes a second for these Labster simulations to load because they're downloading basically like a little video game for you. What you want to do while these things are downloading, you need to stay on the tab that's open. If you navigate away from the Labster tab, it pauses the download, okay? So just stay on that tab until it's fully downloaded. The second thing that you'll notice about the download is that sometimes it gets hung up at about the 25% complete mark. That is absolutely normal. It'll just kind of sit there for a couple seconds, and then it will progress through and it will load completely. So now hopefully you can see here that if you start a Labster simulation and then you get busy, you get called away and you close it out, when you come back, it'll help you to pick up right from where you left off. So it's absolutely fine if you don't have time to finish the whole thing to come back to it later. So I'm going to, instead of continuing from the saved game, I'm going to restart from the beginning of this demo. And here you can see it's loading for me. It's getting a little hung up at the 24–25% mark. And it should clear in just a couple of seconds and finish loading the entire module. Hopefully, there it goes, okay. All right. You can see, please stay on this tab while loading. Loading will pause if you jump to another tab. All right, now it's ready.
Hi there, welcome to Labster. Here you can experience fully interactive laboratory simulations on many different science topics. Today your mission is to help me identify the biological molecule shown in the image. Click Continue to start with your mission.
Okay, so a few things that I want to point out to you. What we're going to do here in a second is a very, very simple little demonstration on how the Labster platform works. So it's going to ask us some very simple questions. But the basic premise of any Labster simulation is that you do some activities and then it asks you to answer some questions. The questions are how I'm going to be generating a grade for you for these Labster exercises, okay? The second thing you might notice is that there's some very annoying music going on in the background. You can control whether you have the music on or off. You can turn the music off here. You can leave, there are sound effects that will happen throughout this thing. If you don't like those, you can turn those off. And the narrator will read to you. You can decide to turn that off as well. I'm going to turn off the music, but leave these two things on so that you can hear what those sound like. The music is a little distracting while I'm trying to tell you guys about this stuff. But whatever your comfort level is, absolutely, you can do those things. Now, the next thing I want to point out to you is that what we have here is supposed to be sort of like an iPad, okay? This iPad guides you through your lab exercise. And the iPad has a lot of information on it for you. It tells you how far you've gone through the exercise, okay? It also tells you your score. Now, depending on the Labster assignment that I give you, it might say something like 400 here, okay? What I will do is, I'm not gonna give you 400 points for each of these Labsters. I will scale it. So whatever % of the points that you get in your Labster exercise, I'll use that to calculate the points that you get for class. So the Labster exercise that you'll be doing this week will be worth 20 points, and then whatever percentage you get here, I will prorate that into the 20 points in the grade book. If that doesn't make sense, you'll work through it, and I'll be glad to show you how that works. Now, I also want to point out for you that on our little iPad, we have several tabs: the Home tab, Theory tab, Media, and Mission. The Home tab is where you're going to live most of the time. It gives you instructions on what you're going to be doing. It's where the quiz questions will be. You can navigate to anywhere from this Home tab, okay? The Theory tab you can get to here, or you'll often see a little button here that takes you to the same place. And what the Theory tab does is, let's say you've been asked a question that you don't know the answer to. The Theory tab gives you some background information to help you understand what you're doing or the concepts behind the question that you've just been asked, okay? Lots of times Labster will ask you questions about things that it hasn't told you about, because Labster wants you to go and find those answers for yourself using this Theory tab. The Media tab, if you click on that, it shows you all of the pictures that are going to be used throughout this simulation. If we go back here, you can get a similar thing by going to View Image. Back. The last tab here is your Mission tab. So your Mission tab tells you the tasks that the lab is going to have you work through. And I'm going to talk more about the Mission tab as we go through this simulation, all right? So let's go back to our Home tab and let's look for some instructions here. It's telling me to click Continue to start with our mission. So let's do that.
Even though you are in a virtual environment, we take lab safety very seriously. Please wear a lab coat before working in the lab. Turn to the right and click on a lab coat.
Okay, so our little artificially intelligent Dr. One here is giving us some instructions. You have to do what she tells you to do before you can move on in the lab. And you can see she's kind of guiding you.
Click on the lab pad for more information.
Okay, so she's guiding us where she wants us to go. Because we're taking so long, she thinks we're confused, so you can click here as well. So if you click on the lab pad, it brings it up. If you click on the background, it pulls it down. So let's move over here in the direction the arrow is telling us to go. And you can see we have lab coats. Click on those.
Labster gives you the opportunity to learn science by using and exploring all types of equipment, materials, and animal models, including the expensive and dangerous ones. Let's use an inverted microscope to have a look at some cells. Click on the microscope workbench to go there and get started.
Okay, so she wants us to go this direction. We're looking for the microscope workbench. So if you hover over things, it tells you what they are.
Click on the Petri dish to pick it up, then place it on the inverted microscope stage.
Petri dish, and you can tell that's what she wants you to pay attention to because of the glowing. So we're going to click on it. Now she wants us to put it there. You can tell because it's
Nice
glowing.
work so far. You can navigate the lab with no problem. Now, let's dive into the cells to see what's inside. You can't do that in real life, but you can in a virtual lab. Behold the machinery of the cell. These tiny structures are called organelles. Let's follow a protein molecule inside the nucleus. You're inside the nucleus, the place where most DNA is stored. In here, DNA is tightly packed into a long thread-like structure called chromatin. Let's zoom in further on the chromatin. You can see the double helix structure of the DNA. Certain proteins, like the one you see now, can interact with DNA. It is difficult to capture a protein and DNA interaction like this in real life. But in Labster you can dive into the cells anytime you want, in order to see many interesting processes. Great, you're back from your expedition into the cells. Now that you have seen what's inside them, what type of molecule is shown in the picture above?
Okay, so this is how you're going to get a grade for this Labster simulation, by answering questions that you're asked throughout. So every Labster question starts out being worth 10 points. And the way that Labster scores you is you've got 4 options here, right? So hopefully everyone knows what this molecule is. But let's say that you answer this question incorrectly. I'm going to click protein.
No, I think this is incorrect. Please try again. Check out the Theory tab on your lab pad if you don't know the answer.
Okay, so it's letting you know that if you're unsure, you can always click here to get background information, or you can click here. Both take you to the same place. So I'm actually not going to click that. I'm going to go to continue. And what I want to show you is, let's go ahead and click a wrong answer
No,
again.
I think this is incorrect. Please try again. Check out the Theory tab on your lab pad if you don't know the answer.
All right. Now notice every time we go back, it's giving us the same 4 options, but it does scramble the order. So we've answered protein, we've answered carbohydrate. Let's do RNA.
No, I think this is incorrect. Please try again. Check.
Okay, so that leaves us with 1 option left, and that option is DNA.
You are a genius. A DNA molecule is composed of 2 long strands that spiral, creating a double helix structure. Your human brain really triggers the evolution of my AI brain.
Okay, now what I wanted to show you is that I scored 4 out of 10 points. So every time I answered incorrectly, it started me out with 10 points possible, and I got it wrong once and it took off 2 points, and I got it wrong twice and it took off 2 points, and it takes off 2 points each time you get it incorrect. So you have multiple opportunities to score, to make a score on this question. You get some degree of points just for trying this thing out, right? So basically I got 40% on this item because I tried, okay? So basically what I'll do when I am generating your grade for one of these Labsters, for example, the Labster that you will be assigned will be worth 20 points. I'll take whatever percentage you ended up with and multiply that times the 20 points to determine how many of those 20 points you get awarded in my gradebook. Okay?
Thank you. You helped me identify a fundamental biological molecule. It was DNA, the molecule that holds all your genetic information. If you want to keep exploring the lab, feel free to do so. You can say hello to our iguana or have a look at what a molecule of caffeine looks like.
Okay, so
Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation?
Okay, it always asks this at the end of every simulation, but before we answer that question, I want to back up. I want to show you this mission tab that I've shown you before. Now, I'm not going to do this right now, but it's got all these different checkpoints, things that we were supposed to accomplish throughout the course of our Labster assignment, and we've done all 4 of them. Now, occasionally, you may find yourself doing an activity where you can't seem to work past it. Maybe your computer has gotten stuck, or maybe, sometimes there'll be puzzles or something like that, and you just need to back up and start over. If that's the case, your best bet is to navigate to this mission tab and then tell it where you want to go. So if we wanted to go back to getting ready to work in the lab where we put on our lab coat, we would click load that checkpoint and it would take us back to that part of the simulation. So you can use this mission tab to go backwards whenever you find yourself stuck on a Labster task, okay? But in this case, I'm going to go ahead.
Thank you for your feedback.
Okay.
Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.
When you get this simulation complete badge, that tells you that Labster thinks that you're finished and that your grade has been populated to the Blackboard gradebook, okay? So you need to work through all of your Labster simulations until you get to this simulation complete page. All right, hopefully you guys kind of were able to follow along with that. I have left this here for you in case you want to play around with it on your own. And then once you feel comfortable, this has got no credit associated with this. This is purely for practice for you. Once you feel comfortable navigating the Labster environment, then you can go ahead and start working on your introductory polymerase chain reaction assignment. Now that I've shown you a little bit about, about the Labster platform and told you something about our online activities, let me give you some background information about the processes that you'll be simulating in your assignment this week. So this week, we're going to go over something called the polymerase chain reaction, which is typically abbreviated PCR. And then we're also going to go over a process called electrophoresis. So let's start out with PCR. What is it and what does it do? PCR is essentially a technique that copies regions of DNA, okay? So it can take a very small amount of DNA and then amplify it into a larger pool of DNA. So you'll often hear of PCR as sort of a molecular Xerox mechanism, okay? Now, one question that you might be asking yourself is, why would we need to make copies of DNA? Okay, so why would we need to make copies of DNA? Well, let's think about the times that you might need to analyze DNA. So for example, perhaps you're talking about a crime scene. Maybe you got like a blood sample, and that's what we'll be talking about this week in our Labster. Or perhaps you have a sample of tissue. Later on this semester, we'll be taking some fish tissue from a local restaurant, and we will extract DNA from that tissue and use PCR followed by sequencing of genes to determine what species of fish you have. So the reason that we need to make copies of DNA is because the DNA that we extract from samples like these is usually not enough for us to analyze, okay? We usually only get a little bit of DNA out of these samples. And so what we need to do is to make a whole bunch of copies of the DNA in these samples in order to be able to analyze it. DNA, you have to have a lot of it to be able to play with it, to be able to see it or sequence it or anything like that. So we have to make copies in order to work with it. And that the copies are accomplished via PCR, all right? So let's talk a little bit about how PCR works. The very first thing that you have to do is you have to get DNA out of your sample. So for example, as I mentioned, if we've got blood, right, you've got to extract DNA from the nucleus of blood cells. You'll learn in this laboratory that not all blood cells have nuclei. Perhaps you already know that red blood cells lack nuclei. But white blood cells do have nuclei, and we can extract DNA from those white blood cells. If we are talking about something like muscle tissue, okay, like when we extract DNA from the fish that we get from a sushi restaurant, we'll be getting DNA out of the nuclei of these muscle cells, okay? So that's your step one. You want to extract your DNA. And we're going to call this DNA that we extract template DNA. It is what we're going to copy, okay? Now, once we have our sample template, we are going to create a reaction. So PCR stands for polymerase chain reaction. It is a chemical reaction. Okay, and so let's make ourselves a little chemical test tube, and let's talk about the things that are going to go into this chemical reaction. The first thing that we're actually, I should have put 5 things on here. All right, the first thing that we are going to put in our test tube is something called a buffer, okay? You can kind of think of this as just sort of the water that things are taking place in. It's going to have ions and other chemicals in it, okay? So I'm gonna put buffer here as our number 1 component of our reaction, okay? And that's this guy right there. All right, so we've kind of filled up our reaction tube with some buffer, or you can think of this as almost being like the water of the tube. The next thing that we have to put in there is what we want to copy. So we're going to put in our template DNA. That's this guy, and I'm going to draw it like this in here. Okay, it's very terrible double helix, but you get the idea. All right, so there's our template DNA. This is what we want to copy. All right, now the next thing that we need are the building blocks of DNA, and those are called nucleotides. Now, here, that's what these are here. Here you see them abbreviated DNTPs. Okay, that stands for deoxynucleotide triphosphate. Now, we'll get into more detail as to why these are called deoxynucleotide triphosphates later. But what I want you to know for now is that these are your DNA building blocks. These are the A's, or adenines, C's, or cytosines, T, or thymines, and G, or guanines, okay? So your A, C's, T's, and G's, or your building blocks, all of these guys are going to go in here. Because if we're gonna make copies of DNA, we've gotta have the building blocks of DNA, right? So those are all going to be floating around in there. Now we have to have some sort of machinery that's capable of making new DNA molecules. And that machinery is an enzyme called DNA polymerase. Okay, and it's actually a very specific kind of DNA polymerase that I'm going to tell you about in just a second, but it is an enzyme, and what it's going to do is it's going to take all of these building blocks and assemble them into a copy of our template here. The last thing that we need are some little tiny starter pieces of DNA, and I'll talk more about these in a second, but these are called primers, all right? Primers are small stretches of DNA. They're usually about 20 base pairs long, okay, give or take a couple of base pairs. And these are going to be important in identifying the region that we want to copy, because we typically don't copy the entire DNA molecule. We're looking to copy a small part of the DNA molecule. And so the primers help us to identify the region that we want to copy, so that maybe we're only copying maybe from here to here or something like that. Okay, we don't need to copy the whole daggum molecule. All right, so those are the ingredients for our PCR reaction. Now, how does PCR work? All right, the PCR happens in a few steps that I've summarized for you here, but we're going to walk through them. So I'm going to draw a hypothetical. Well, maybe I am. I don't know why it's not drawing. Hold on. Give me a second. There we go. I am going to draw a hypothetical template strand of DNA here. Now, at this point in your studies with Dr. Bauer, I am going to assume that you know a little bit about the structure of DNA. So you know things like it is double-stranded, it is in a double helix, it is anti-parallel. So you can see here, I'm drawing for you 2 strands. I'm not really drawing them as a helix, but I am drawing them as being anti-parallel with 5' at one end, 3' here, and then the opposite being true here and here. Okay, now remember from your lecture that these 2 strands are being held together via hydrogen bonds, aren't they? So the first step of PCR is going to be to make copies by first splitting these 2 strands apart. And the way that we're going to do that is we are going to apply heat. We're actually going to heat this reaction up to about 95 degrees Celsius. Now, remember that in Celsius, 100 degrees is boiling. So we're almost going to boil this DNA. And when we do that, when we heat things up, we're increasing the molecular motion, so that what's going to happen is the hydrogen bonds that were holding these 2 complementary strands together will fall apart because of the molecular motion of the increased heat. Okay? So now we have pulled these 2 guys apart by heating them up. This step is called the denaturing step. Denaturing is when molecules fall apart. So hopefully you remember that proteins can denature, but so can DNA, okay? The denaturing step, that's step number 1. Now, in step number 2, what's going to happen is we're going to cool this reaction down a little bit. We're going to cool it down, and depending on, different reactions get cooled down to different temperatures, but usually somewhere around 55 degrees C. And what that does is it slows down the molecular motion of the molecules in our reaction mixture. And what it's going to do is it's going to allow those little primers that I talked about earlier, they're going to be moving slow enough now that they're going to be able to line up with our template DNA. So what these primers are is short little segments of DNA that complement our templates. I'm going to actually draw these in a different color. So here we have a short segment of DNA, and it's going to have complementary base pairs to this region here. And we've got a short segment of DNA that's going to have complementary base pairs to this segment here. Now, why do we use these primers? Well, basically what's going to happen in just a moment is DNA polymerase is going to grab on to these regions here and here. For reasons that we will talk about when we talk in more detail about PCR and about DNA replication, DNA polymerase needs 2 strands to grab onto. It needs 2 strands to grab onto. Once it grabs onto 2 strands, it can start building copies for us in this direction or this direction. But it needs those 2 strands to get the party started. So in order to establish a double strand here and a double strand here, we use these short little segments called primers. These primers are able to line up with their complementary sequences because we've cooled our reaction mixture down and we have allowed the whole thing to stop moving so fast due to molecular motion. So we call this step here the annealing step. Now, after our primers have annealed, and let's see, I'm running out of space. Let's see here, where can I draw this? I'm gonna draw it up here. After our primers have annealed, so we've got 1, 2, we've got our primers, boom, boom. The next thing that's going to happen, I kind of mentioned it here, the next thing that's going to happen, and let me do this in green, is that our DNA polymerase enzyme is able to grab on to these double-stranded segments and it's going to elongate a copy in this direction and a copy in this direction. So you can see that here we had our original and we're going to make a copy so that this will be a double-stranded molecule. Here was our original. Now we're going to make a copy. Now this is a double-stranded molecule. So where we started with 1 copy, we're going to have 2 copies. Okay, hopefully you can see that. This happens, we're going to heat this, actually this happens when we heat this reaction mixture up to 72 degrees C. Now why are we heating this reaction mixture up? DNA polymerase works best at this temperature. The type of DNA polymerase that we use is something called Taq polymerase, and I'll be talking about that in more detail later. It is mentioned in your labster. This step is called the elongation step. So let's review these steps one more time using the figure over here. And let's see, I'll do this in purple so we don't get confused. Okay, so here's our original template. We're going to heat this template up to split these in 2, okay? So this is our denaturing step. After we split these in 2, we're going to have those little segments of DNA come along because we're going to cool this reaction mixture down. That's going to allow these little segments of complementary DNA to line up at the beginning of the regions we want to copy. This is called the annealing step. When you anneal something, you bring it together, okay? Then what's going to happen, we're gonna heat this thing back up to about 72 degrees C. We heat it up, DNA polymerase is able to bind to the double strand here, binds to the double strand here, and it starts synthesizing a new strand in this direction and a new strand in that direction, okay? So what happens, this is the elongation step, What happens is we started with one molecule of template DNA. By the time we finish our first cycle, we have 2 copies. We've doubled the amount. And so you would repeat this process over and over and over again. And with each new cycle, you are doubling the amount of DNA copies in your reaction, okay? And you'll do this over and over and over again until you have sufficient DNA to do an analysis. Now, in our simulation, in our Labster simulation this week, we are going to be copying a very specific region of DNA, okay? We are going to be copying something called tandem repeats. Now, what in the heck is that, and why would we want to copy them? Well, let me back up and tell you a little bit about the premise of the lab that you'll be doing this week. In this Labster simulation, you're investigating a murder. And what you have is a professor who's been murdered, and there are 3 suspects. There is a rival scientist, okay? There is a PhD student, and there is someone called a postdoctoral fellow, or postdoc for short. So this is someone that's gotten their PhD and they're doing some extra work after their PhD. So one of these 3 individuals has murdered a professor. We don't know who it is, but we do have some of the murderer's blood from the crime scene. So here's what you're going to use PCR to do. Notice that we have these 3 individuals, and here's some of their DNA here, visualized here. The blue regions are identical. So this blue region is the same in this individual, and this individual. This blue region is the same here, 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 here. But notice that these regions are a little bit different. What these regions are is areas where we have a short sequence of DNA. So this one's AGAT, okay? This sequence is repeated in this individual 7 times. 1, 2, 3, 4, 5, 6, 7. So it's AGAT 7 different times. This person has AGAT 1, 2, 3, 4 times. This person has it 1, 2, 3, 4, all the way up to 12 times. These areas where you have a sequence that's repeated over and over are called short tandem repeats. So they're short, AGAT. Tandem means one right after the other, and they're repeated. What's interesting is they're repeated different amounts in different individuals. Why is that? Well, we think that that's because during DNA replication, the DNA machine, the cellular machinery that copies your DNA gets confused when it sees a bunch of these back to back, and sometimes it kind of hiccups and it makes more copies than it should. So it sees AGAT over and over and accidentally copies it too many times. And so you end up with variable numbers of these short little sequences in different individuals. Now, here's the neat thing. We can use PCR to analyze this information. What we do is we put one of our primers on this side of the repeat and one of our primers here, one of our primers here, and one of our primers here, here, and here. Because remember, this DNA is identical and this DNA is identical. So now what's going to happen is after PCR, we're going to copy all of this. After PCR, we'll copy all of this. After PCR, we will copy all of this. What I hope you can see is that the PCR product we get from this person is going to be a very short strand of DNA. The PCR product we get from this individual is going to be a medium length strand of DNA. And the PCR product we get from this individual is going to be a long strand of DNA. So we can use PCR to amplify these short tandem repeat regions, or, oops, come here, oops, stop it, sorry, or STR regions. And we can determine how long these regions are. We can then use PCR on the murderer's blood and see how long that region is here. Compare the length of this to these lengths to tell who's the murderer. Now, how are we going to determine the length of our PCR product? That's where the second process comes in. We are going to use something called gel electrophoresis. This is going to use electricity, okay? Gel electrophoresis is a very simple process. Basically what you do is you're going to make something that's almost like a Jell-O jiggler, okay? You're gonna make a square of what looks kind of like Jell-O. It's actually not gelatin, it's a substance called agarose, but it's very similar to gelatin, okay? So hopefully you can see that I have a block of gelatin here. And again, this is made of something called agarose. Now, in this block of gel here, hardened gel, we're going to have actually let me put 1, 2, 3, 4, 5 divots, okay? Make this smaller. 1, 2, 3, 4, 5. Okay, these are like little holes. What we're going to do is we're going to put our PCR product in these holes, okay? So we're gonna put our PCR product from the rival here, our PCR product from the PhD student here. We'll put the PCR product from the postdoc here, and we'll put the PCR product from the murderer here. What we're going to do next is we're going to run an electrical current through this gel. And we're going to have the negative end of the electrical current on this end and the positive end here. Now, why is that? Well, it turns out that DNA, which is what our PCR product is, right? It's DNA. It is negatively charged. So by running a current going negative to positive, what we're going to do is we're going to repel our DNA, and it's going to move through this gel, through the inside of this gel, and it's going to be attracted to this positive end because the DNA itself is negative. Now, what's interesting about this whole process is if we could cross-section this gel, if we cut it in half and look at a cross-section, so now I'm looking at a cross-section here, and we could look at it really up close with the microscope, what we would find is that it is actually composed of a series of fibers, right, that make like a mesh. So it's almost like the inside of a sponge. So as the DNA is moving through this gel, it's got to move in between these fibers, right? Big pieces of DNA get caught in these fibers and they move very, very slowly because it's hard for them to progress through this fibrous matrix. Small pieces of DNA are able to navigate through these fibers more rapidly because they fit through all these little holes. So if you have an individual who had a short PCR product, it will move really fast and it'll move down toward the bottom of the gel. And you'll be able to see a little band like that. If you have a PCR product that's intermediate, it'll kind of move in between. It'll be kind of in the middle of the gel. And if you have a PCR product that's really long or really big, it's going to move really slowly. So it'll be more toward the top of the gel. So you'll see little bands of DNA, and the farther the DNA has moved, the smaller the fragment is. So let me go back. This individual, I think I may have it wrong on my gel, this individual has the shortest DNA. You would expect it to migrate the farthest. Now, we can also load up the murderer's DNA. So let's load it up. Depending on where it falls out, you might be able to tell who the murderer is. So in my hypothetical example, I've got the murderer DNA being at kind of an intermediate level, and lo and behold, it's matching this individual here. So this individual is the suspect for our murder. Now you may be wondering what's going on in this lane right here. Why? These are called lanes. You can kind of tell this is a lane, this is a lane, this is a lane, and this is a lane. All right, so I have a well here and a lane here that I haven't used yet. What we typically load into this lane is what's called a DNA ladder. And what that is something that you buy commercially, and it consists of a whole bunch of different fragments of different sizes. And it basically acts as a marker. So like, for example, this DNA ladder, maybe it tells us that this particular fragment is, I don't know, let's say that it's 50 base pairs long. So this gives you some idea of how big. Maybe this one is 100 base pairs long. And maybe this one is 200 base pairs long. So these DNA ladders give you some idea of how big your PCR product is. So this is just a little brief introduction to help you understand the Labster simulation that you're about to do. I will be going back over all of this in our in-person laboratory. So if you're a little confused, don't worry. All of this will become clear as we move through the course. I have just a few more things to tell you before I let you go. As you're working through the Labster simulation, make sure that you read those theory pages. Those theory pages are going to tell you most of what I just told you. They're going to give you a lot more detail, and they'll also have some lovely images and illustrations for you. Don't forget to do what the Labster simulation asks of you. If you don't put on the lab coat like it wants you to, if you don't put on the gloves it asks you to, if you don't click on the glowing objects, then it's just going to get hung up and you won't be able to progress through the game. The last thing is you're going to use something called pipettes. It's going to ask you to put together a simulated PCR reaction, and it's going to ask you to dispense volumes of various things like nucleotides or DNA template. And you're going to use something called a pipette to do that. Make sure that as you use the pipette, it's going to ask you to pick up a new pipette tip each time you use the pipette, and it's going to ask you to discard the old tips. It will direct you if you do things incorrectly, but I just want to sort of cue you into this so that you're aware if you for some reason can't progress, you might want to play with where to pick up tips or where to discard tips in your simulation, okay? If you have any questions about this simulation, please let me know. Remember, you have some time to get it done, and I'll be happy to answer any questions and help you in any way I can. Good luck, and I look forward to working with you this semester. Bye-bye.